Determining a patient's predisposition to heart disease often involves examining which form of a specific gene, which alleles they possess, because certain alleles may predispose one towards disease. Now, while patients do provide DNA in cheek swabs and blood samples and the like, it is not enough DNA to perform these tests. And so a technique known as polymerase chain reaction, or PCR, is used, in which a small amount of DNA is amplified to make sufficient quantities to test. There are two secrets in this process. As DNA is heated, the double-stranded molecules separate, and then new complementary DNA molecules are created, and then the process is repeated. The enzymes used originate from bacteria which are tolerant to extremely high temperatures. And this way, new enzyme does not have to be repeatedly added. This was originally done and it was more costly. Secondly, one can control which portions of the human genome are amplified by using specific RNA primers. The DNA replicating enzymes, DNA polymerase, cannot bind to DNA without binding to a primer first. And by using specific primers, one then controls which segments of DNA are replicated, namely the segments involving the gene that one is analyzing in a patient. So PCR is the technique which amplifies a small amount of patient DNA to give sufficient quantities for testing.